So greetings to all from Tunisia this time. And we are attending the last seminar for 2023. So this is the 8th seminar in the series of Crops to Hunger, case studies in Africa and beyond, supporting CGIAR partners through genotyping services. So thank you for your time and your presence. So in our previous seminar, we had Gloria Adu from Ghana, who presented us with very interesting updates on the breeding and genetics of maize in our institute. And today we have with us Dr. Brigitte Uwimana from the IITA Banana Program, who will be introduced by Dr. Alan Brown. Thank you very much for both to accepting this challenge. And Dr. Alan Brown is a plant breeder who obtained his PhD from the University of Illinois and has a great experience with both conventional and market-assisted breeding of several vegetable and fruit crops, but also with maize. At North Carolina State University, he was the lead PI on an international collaborative project to generate the first genomic draft sequence of blueberry, and was the first researcher to use high-density SNP arrays of rapeseed to generate saturated genetic linkage maps of related plant species. He holds the title of Emeritus Professor of the University of California, Riverside, and currently he is the banana breeder at IITA Tanzania. Thank you very much, Dr. Alan, for accepting this. So today I can't show, but we are here in the audience around 25 persons because we are in a training for genotyping in Tunisia. So it's not just 26 people that are in the online, but also around 25 people that are here willing to listen you. Dr. Alan, please go ahead.
Thanks, Anna, for that very, very nice introduction there. Only one minor correction. I'm not Professor Emeritus from UC Riverside. So just one minor little detail. Good afternoon, good evening, everybody. It's a pleasure for me to introduce Brigitte Uwimana, who is the molecular breeder for IITA Banana in East Africa. Brigitte's been with the banana breeding program now for 10 years. The last 8, she has been stationed in Uganda. She is originally Rwandan and has been doing some wonderful work for us there in Uganda. Banana breeding, as you can imagine, is a challenging endeavor. You're starting out with a plant that's almost completely sterile. You're going through enough recombination in order to produce an improved variety over what you currently have. And then at the end of the day, you have to have also a seedless fruit. So it presents a number of challenges. Having said that, however, the banana breeding program has made some great progress in recent years. We produced the first Matoki cooking banana hybrids that were released in Tanzania and are soon to be released in Uganda. We have produced the Machadi cooking bananas here in Tanzania that are approaching release. So we have made some very good progress here. But of course, just producing hybrids and releasing them is not the end of the story. You have to have the material get to the farmers, and you have to be able to track the impact that this material is having on the local economy. And that's where some of Brigitte's talk will come in today. Is you have 2 problems with banana. First of all, it's a clonally propagated crop. So people are not buying seed. They are going to their neighbors and they're getting clones for the banana. So it's hard to keep track of banana as it moves into the distribution chain. It's also a bit of a problem because the names change very frequently. You might release the name of a particular variety, but as it goes to the farmers, they will adopt new names for this variety. You might call one banana a certain name in one part of the country, and then 50 kilometers down the road, the same banana will have a different name. So it's hard to keep track of this material, and it really requires the use of molecular markers, which Brigitte will talk about here in her talk. Again, I want to thank Anna for the introduction, and I want to thank everybody for joining. And if Bridget, if you're ready to get started, I'll turn it over to you.
Thank you very much, Alan. Let me share my screen if I remember how to do it. Can you see my screen?
Yes. Just put it in presentation mode, please, Brigitte.
Yes. How do I change this? Which screen are you looking at? I have two screens here with me.
Not as the presentation mode, Brigitte.
Is that better?
Yes.
Okay. Okay, good afternoon, good morning, everyone. I'm going to present to you the work we've been doing on molecular breeding, but I focus mostly on the work we are collaborating on with the former module 3 of AIB, now part of BRI. The people who have been active involved in this work in IT are myself, the Trusha Shah, the bioinformatician based in Arusha. There is Ivan Alinayitwe, the molecular breeder from the national program here in Uganda, Delphina Ma, who is the plantain breeder in Ibadan. Now the slides are not moving. They are moving. So as Alan talked about briefly, most of them hear about bananas. You understand Cavendish because that's a commonly known banana. But at IT, together with the partners, we don't focus on desert bananas, but we focus on cooking bananas. And we have 3 types we breed for. What we call matoke. So these are the cooking East African iron bananas. And the mshares, as I mentioned, these are triploids, AA. Then the plantains, many people know plantains. So the difference between the 3 is in the haploidy levels, but also in their genomic composition. As you can see that plantain has an additional genome, which we call the Barbizena genome or the B genome, while the others are A-based, which is Musa acuminata. There are many different structures in their use because the products they give are totally different in terms of consumption. You can't give a mshare to someone from Uganda and tell them to cook it as a matoke. It will not work. The same with plantains or any of these in the different geographical location. So the matoke and mshare are mostly in East Africa, but the plantains are more abundant in Central and Western Africa. The target product profiles for the 3 types of bananas have something in common. They share the same pest and diseases mostly, but we see differences as we move through my slides. The agronomic traits, they don't look the same, but farmers still want a big bunch. They want no seeds in their bananas. And the bunch orientation is the way the bunch is dependent on the pseudostem, which means if it's facing up, which we have sometimes, it does give a good bunch. Plant stature is a translation of the ratio between plant height and plant girth, which translates into the strength of the pseudostems to sustain a big bunch. Now, when we talk about the 2 types of traits, pest and diseases and agronomic traits, they are target to be improved. That means that we need better yield, we need resistance. But for the consumer's traits, when you want to improve them, that's a suicide mission. So what we do, we maintain the quality that the farmers or the consumers want. So that's the target. So we move into the KASP markers for different purposes. But before we go there, as Alan mentioned, or as I even mentioned in the last slide, the final products of the mshare, of the matoke and plantains are supposed to be triploids, but those from mshare, they can be triploid, but the target is them to be diploids. So the most SNPs we all know, they look like this in diploids, but when you go into polyploids, the 3X, we look like this. We have 2 additional groups at the heterozygous state. And when you go to tetraploid state, we get 3 classes of the polyploids. This has meaning in the banana breeding because that tetraploid are intermediate products which help to increase the seed set, but most of the final products are supposed to be 3X, and the 2X's are used as parents. A bit complicated, but I hope you understand. So what this Zoom is covering is these are KASP markers for fruit filling. What we call fruit filling in the bananas is the difference between this bunch, and this bunch. This is what you would hope to get in your field, but as a product of breeding, we get some of this as well. So if you can't eliminate this at an early stage, that means you have to wait at least 4 years to be able to get rid of this, which means that you have wasted a lot of resources, and also you have wasted a lot of time when you could have moved on with better products. So the fruit filling basically means fruit diameter. It means pulp diameter. It also means fruit circumference. Why is that so? We measured the 3 parameters, and we found that they are highly correlated as it would be expected. So the correlation is as high as 0.99, which means it will be easier to measure fruit circumference because you don't need to cut. But if you have time, you can do this, but it will be easier just to measure fruit circumference. And these 3 parameters are highly correlated with bunch weight, which is the main parameter for yield. As you can see the correlation here, which is at 0.4 up to 0.81. So if you can improve fruit circumference, you are automatically increasing yield for your bananas. And these traits, which are highly correlated with the bunch weight, they have a very high heritability, this broad center heritability, which is almost close to 1, almost the perfect heritability that you would expect. So QT analysis for these traits was run in matoke background GWAS population. And a major QT was identified of chromosome 3, as you can see here. And there were some markers which were in the anchored cortex of the Pahang genome. But further analysis identified that these same markers also in the QTL region. So we could follow this QTL without any fear because this was not representing additional QTL. The significant markers for the QTL were shared by, they were shared to Intertech together with the surrounding SNPs, 50 upstream, 50 downstream for the verification, for the development of the assay for KASP markers. And out of the 37, 24 were successful, and these were validated. Now in the validation process, remember these are the final products. So as we get the calls from Intertech, they come as 2X. We had to use FitPoly to call the genotypes as triploids. As mentioned before, you can see that FitPoly can identify the homozygous for one allele, the homozygous for the other allele, and the 2 groups of the heterozygous, which helps to understand how the QTL works in terms of allele dosage. This is an example of 2 markers out of the 24. As you can see, 1 allele of C for this particular SNP is enough to increase pulp diameter. So when I talk about pulp, again, is this part of the banana which is eaten. So when you have pulp, it's this one. So a pulp diameter of 1.5 is almost centimeters, almost like something we throw away. And this pulp diameter here is something we want to continue with. So as you can see, one increase in 1 allele is enough to increase from 1.5 to 2.5 or to 3. As you can also see that the allele dosage is there, but based on this data we have, there was no significant difference between the 3 dosages of, allele T is one which increases the phenotype. Here, the homozygous GG is the poorest, and 1 dosage of C is enough to increase from this to up to there. And you can see the p-values. This was based on ANOVA on each marker, and the power of detection is a depiction of how good the 3 classes can be differentiated if you have enough genotype classes to identify, to differentiate between the 4 groups. And the heritability was also calculated for each trait, for each marker. So based on this, these markers are now being applied in 4x by 2x crosses. These represent stage 3 of the breeding, banana breeding. And this is the equivalent of the stage where the products are being developed before they go to further evaluation in the field and on farm. And based on these markers and what we are now, the application we're seeing now, we are able to eliminate 40 to 6% of the genotypes, which are now thrown away without going to the field. And you can move on with your genotypes. So we evaluated, we genotyped the other backgrounds because this was mapped in Matoke. So the first background we used was the Mshari background. Remember, Mshari is AA, diploid. And we saw the same trend, but this was only based on 53 genotypes. You can see again that with 1 allele, you see an increase in the fruit diameter, but this was based on a few genotypes. We have already genotyped the next ET, which has about more than 200 genotypes. We are waiting for the phenotypic data. We have also genotyped the plantains in terms of the PYT and ET, which is a mixture of poor and good fruit feeding, and also genotyped the 2X. We are waiting for the phenotypic data to be able to see how this QTL behaves in other genetic backgrounds. But also these same trials also help us to map for the same traits in the other backgrounds. For pests and diseases, we go through a few of them that I've worked on so far. This is a disease called banana xanthomonas wilt. It's common in East Africa and part of Central Africa. It's not yet in West Africa and not yet also in Arusha, where the msharis are grown. The peculiarity of this disease is that once it affects a plant, you get 0 yield because you get this kind of fruits which you can't eat. The screening of the scoping for the different genotypes for this disease identified few Musa acuminata which were partially resistant, and these were mostly in the subspecies of Zebrina. Barbeziana is very resistant, but the use of it will require a lot of pre-breeding to get it to a point where it can be introduced into the parents, because the bee genome comes with a lot of unwanted traits, especially for the Musa acuminata genotypes. So the QTL for this particular trait were mapped in the population called Monyet Kokopo. So Monyet is the partial resistant one, and Kokopo is susceptible. And this project, because from 4x by 2x, it's a triploid. We used a system called a continuous mapping of QTL, which is about regressing the allele frequencies to the phenotypes instead of coding. And this was for two reasons. One is because as a triploid, we needed very good sequences to be able to code as a triploid, which were not available. So with DATSIC data, this was the best option we could get. So we found about 5 QTL for this particular disease, which were based on AUDPC. And the significant markers were again shared with Intertech for the development of KASP markers. We got the results not long ago. So we're analyzing these ones and they're in the pipeline. We also mapped QTL for weevils, which is again the biggest pest for banana and plantains. This one is shared throughout wherever bananas have grown for a long time. And the yield loss for this one is up to 100% over time, because if this disease, this pest was not around, and if you would have any nematodes as well, you can have banana plantation for 20 years. But with this pest, you can't expect harvest longer than 5 years. So the effect of the pest is the damage on yield, which gets worse over time as you get more inoculum in the field. So here we are talking about QT analysis, again, the monyet cocoa population. And we were using 2 traits, peripheral damage and cross-section damage. And we identified these SNPs which are associated with the trait. And 44 of them were associated with peripheral damage. And 11 of them were associated with cross-section damage. And they have one particular SNP, one particular QTL here at the distal end of chromosome 6. So again, these are 55 KASP markers that have been recently verified. We are going to work also on validating them in the coming year. Fusarium wilt is another disease of bananas. Plantain and matoke are resistant naturally, but if you introduce parents which are susceptible, you can make the final product susceptible. But mshare is very susceptible to both FOC1 and TR4. This TR4 particular strain is not available enough in East Africa yet, but it's the biggest threat worldwide because it's spreading very fast. And in Africa, it's been so far spotted in Mozambique. And surveillance are on with the pathologist to see if it's going up to reach East Africa. So because this one is not available in our sessions, the phenotyping is done with our partners, mainly Wageningen University, Kijin, the University of Stellenbosch, and Queensland University. The QT we're talking about here was mapped in the Maraxense background, which is one of the banana progenitors. And we identified the QT on chromosome 3, and the 24 markers were just being verified as I'm talking. And this is again for the validation for next year. We have developed markers for quality control and quality assurance, both in the, we started with the A genome actually. So we started with 18 genotypes, which make the core diploid breeding population for East Africa. And we got about 5.5 million SNPs. And from there, we selected 55 SNPs, which were representing the 11 chromosomes with 5 SNPs per chromosome. And these were verified in Matoke and Mshare. And the results were good. They had a very high reproducibility, and 29 of them worked well. And we also tried them in plantains, but in plantains, they did not work well because, especially the B genome, which were best tested on the Balbisiana, we only got 13 KASP markers, which amplified. So the next step was to develop this specific SNPs for QCQA, specifically for plantains. And that was done as well. We based the SNPs on the plantain genome, which was the novel genome sequencing by the Institute of Experimental Botany in the Czech Republic. And looking at the super scaffold of the A and B genome, focused on those which were mapping only on the B genome. And again, we went with 55 markers. And again, the results have just been obtained. We are going to be verifying this in the coming months. But with A genome QCQA markers, we have done some applications, and one of them was to test if the genotypes we have of the same names from different stations are actually the same. Many of them were the same, but some were not. This is an example of Calcutta 4, a famous genotype, which is a donor for different pest and disease resistance. As you can see, this genotype in Sendusu, these two samples from Sendusu are the same as the samples from Arusha, but the one in Ikawa is totally different. So this got us thinking, if this one is like this was just a random match, how much differences do we have in our parents? So the project we're submitting actually this week with Crop to End Hunger is to genotype all the parents that have different stations, focusing on just the 3 mats per station, and this mats have tagged. We are hoping to get less differences, but if we get a lot, whatever we find is the same as what has been screened but in the pathology, we want to continue with. We also tested the markers for F1 verification in diploids, and this was the diploids which are bred in Ibadan. We also did the same for the diploid at the national program. And we identified some genotypes which were belonging to the right parents as they are assigned. This is important in bananas. I explain why. Bananas, when you are crossing, you cross one same mat 7 days, which means one same bunch, which means you come today, you pollinate, you go back, you come the following day, you find other female flowers which have opened, you pollinate them again, you go back, you come back, you see the potential making errors. So if there is a mistake in the parents you are attended, or if there is a mislabeling of the parents in the field, it's very easy to get the genotypes, the hybrids which are not from the parents you were hoping to get. So we test these markers for this purpose, and the markers work well. Although one problem we had is that in some diploids, the amplification, some markers were not amplifying, as the maximum of markers we got was 18, especially the diploids. We also tested these, the markers on impact study. As Alan mentioned, the only way we can track the impact we are making is by fingerprinting. So there was a project by SPEAR, which was working with different crops in Uganda, including bananas. So we subcontracted to handle the genotyping of the banana specifically. And 2 genotype platforms were used, DATSIC, but used the same opportunity to test our markers. So the results you see here are for the reference library of 133 genotypes. What we saw was, as it was expected, you can see a list of markers, accessions here, which are not differentiated. So these are Andres Matoke, and others here, these are Andres Mshare, and others here, which are also some synonymous genotypes, especially in the Cavendish. So the library worked well, and we tested on the unknown genotypes, which are about 2,000 3,000 something of the unknown genotypes. And what we identified is that 73% of the genotypes in the area they had sampled were land races, and others were the genotypes which have been introduced. And we noticed 4% of the hybrids which had been released. I'm not at liberty to share the results yet because this was a project by CGIAR. They have not released the results yet. Now, based on the way these markers can work well, we are working on streamlining them in the breeding process. This is for the improvement of parents, which are many diploid and tetraploids. This is the process of the crosses. You start with the field where you do pollination. The plants go to tissue culture. They go to the nursery. From the nursery, they go to the field. They can also at the same time go to pathology screening. Then you get your results that you can recycle to further evaluate, to further crossing, or if you get good parents, you can add them into the 4x by 2x process. So we identify the key areas where errors can happen or where they can be detected for 3 purposes. 1 is F1 verification. 2 is identifying establishing the identity of the parents, which can be done at the same time F1 verification. So after you have the fingerprints, now when do you need to make sure that what you had at the beginning is what you have at the end? The other point is at field screening or pathology screening level, where you don't need to genotype everything, but you need to, what we can do is to do collect samples on each plant and keep it. And if there are specific plants which are acting not normal as outliers, we can genotype them, targeting specific use depending on the results. So what we see here at the nursery level from tissue culture, we can establish the identity of the new hybrids or the new the products of crosses. Then here we do leaf sampling in case we might need, but at a recycle time or at advancement time, we need to confirm again that what was identified here is what is being advanced. In product development, again, this is the whole process. This is where identified that we need to do again the 3 processes, where after selection from EET, you establish the identity of what you are selecting and also confirm that what you have, you are establishing the fingerprints, also the parents of what you were selecting. At PYT, you need to confirm what you are selecting, but also in the field, we can also do leaf sampling on specific parents. And again, before releasing, you need to confirm that what you're releasing here is what came from your initial cross. This is a summary of the samples I've worked on so far for different purposes with the Crop to End Hunger project, the former EIB. So for verification, we have sent this number of samples. You can see that QAQC samples are mostly from the CGIAR project. And in total, we have worked on so far on 6,956 samples in the last about 2 years. The challenges we had, as I mentioned before, calling the diploids was a big challenge at the beginning. We didn't know what to do, but we got the help from the Tools for Polyploid platform that we learned about FitPoly, and we're able to call 3x, 4x as budding dosage. And it's very handy, but it's also extra time, and it takes someone who knows what they're doing, which means it gives more load work on the molecular breeder. The other challenge we have, the Intertech platform works better if you have a lot of samples. You pay really less. But in banana breeding, the number of hybrids you produce per season is not that much, depending on the pipeline you are looking at. So what I've been doing so far, we pull the samples across stations, which means I might send 2 plates from Ibadan, 2 plates from Arusha and 4 plates from Uganda, which combines Naro and IT. Sometimes it works, but sometimes it gets difficult as well. The other thing we need to do, which is we need to work with the whole team, is to streamline the QA/QC markers in the breeding process. So I've talked a lot, but basically I gave you a summary of what we've been doing on marker selection food feeding. That gave the list of other KASP markers we verified for different traits. And we are working on streamlining the use of QC/QA markers in the breeding process. This is my small team. Most of us are here in Uganda, and one person was in Mbarara. This one is based in Arusha. We get a lot of help from the breeding team and the pathology team, especially with getting the material, with the phenotyping work. And we also got a lot of help from Eva and Jaroslav for the de novo sequencing of the plantains. These are different partners we work with, especially with QTL analysis, but also BTI on the Musa base management, because everything works better when we use Musa base. As we get out of files on our computers. So as I mentioned, all this work was funded by ABI, BRI, through the project, the Crop to End Hunger. Thank you very much.
Thank you, Brigitte. Thank you, Alan. Thank you. So the applauses that you are listening is from the 25 people that are here in the training of genotyping. So thank you very much, Brigitte, once again. And the floor is open for questions. So please unmute yourself and welcome questions. In meantime that we are waiting, Brigitte, I think I have 2 questions. So the first is, what is the biggest challenge that to design the KASP markers in Musa? Can you let us know some of your challenges that you faced?
Designing the KASP markers is not that difficult once you know the SNPs you are looking at. But the technical issue of designing is not a problem, but which reference genome should we use? So far I've been using double haploid per hang, which is the best so far the novel sequence of banana. But now we are having more reference genomes coming in, and we will be deciding, because it's better to use the right background than use anything you have. So the best I would actually to use are the parents we have. Now we're having some new sequences coming in, which are based on our parents, which we give more meaningful seeds.
Hello, Brigitte, can I ask a question?
Yes, Rajneesh, please go ahead.
Yeah. Hi. So, Brigitte, thanks for this really lovely presentation. Really updated the research, and really you develop a lot of markers for different disease and also the yields. I have 2 questions. One, when you are doing the QC, what about the plantain? How it works for you? Because we have done the high depth sequencing when we try this with the gene bank material to look, it's duplicate and something. It looks that these markers are not working very well, because majority of the lines saying they are duplicates, but in the field they are looks different. For me, this I face feeling here at my place for plantain. Banana is working well. So that's question 1. And next, yes, yes.
Can you repeat again, like the duplicates and sequences, or what
you are saying QC is working to identify tracking the ID. So I'm saying how the QC markers are working for the plantain case. They are working very well, or most of the time they say, they are the duplicate. Because as I understand, there is a very low diversity in the plantain. So sometime, I don't know, so less number of the markers, they are working well or not. That's I want to know from you. One. And next question is again on the QC. When you are making your QC for everything, then how did you select from the 1,000 of the markers, only few markers that you want to apply only for the developing QC plate or whatever, the QC marker set? So what were the criteria to identify those markers? I'm taking only these markers from genome wide, and from even even on the single chromosome there are the thousands. So how did you select some 4 or 5 from each chromosome? What was the criteria to select them?
Thank you, Rajanish. For the first question for plantains, the markers we have did not amplify well in plantains. From what we saw is that the amplification went for the A genome, but for the B genome, the amplification was not well. But again, from what we know on the molecular side, the plantains are genetically identical. There are very little dissimilarity among them, which is like
Thank you.
0.2.
Thank you very much for confirming me, whatever I'm feeling. Thank you. Yeah.
Yes.
Yeah.
So the target for the QCQ markers is not to differentiate the plantains, but to differentiate the plantain hybrids.
Okay. So you mean there were the QTLs, am I right? Or they were, I mean, or did you select from some, like from the genome wide, and then you just changing the KASP markers? Or they were
Yes,
only the yeah.
The QCQ markers were not based on any QTL. They were based on sequences. And the way they were selected, we had about 18 genotypes, which were sequenced at 30x. And the sites were selected based on 2 criteria, that the coatings or scaffold where their sequences weren't duplicated on the genome, that's 1. And 2, that they're far apart, that there will not be any recombination among them. So that's how they were selected. And the 3 also, they are supposed to be showing was supposed to be showing a lot of variability in the 18 genotypes.
No, I'm fine now. I'm fine.
So that's how they selected.
Yeah, thanks. Now I got it. I was thinking you take it, you are taking it from the DART-seq data from these SNPs and then are identifying, but now I'm clear you are taking it from the sequencing
they were
data.
not based on
So that is
data.
fine. Perfect. Thank you very much. Yeah.
Welcome.
So any other question? So Catherine, you make a comment and you add the link of the banana genome. So I don't know if you want to unmute yourself and make any comment. Otherwise, any other question? And if not, Brigitte, my only question that I have
Anna,
more. Yes,
okay,
Rajneesh.
go ahead. I want to ask one more if the time is there.
Okay, so you can ask, please. Go ahead.
So, Bridget, thank you very much. So I have seen you are showing some QTLs and they are really working very well. And when you are transferring it, then how much it is going to contribute in the line? Because sometimes we are getting the QTLs, they're saying the impact is going to be this much, but we know that's only single QTL. Yeah. But when the QTL is transferring, then interaction is there, environment is there. So how much difference you are getting in the whatever you are looking on, I mean, single QTL based impact, and then in the field, I mean, how much is going to be increased? Like it's saying 1.5 times increase, but in the field, how much it is going to increase in the reality? Do you have any experience on that one?
The QTL, this particular QTL that on fruit feeding was based on 3 trials in 2 contrasting sites. And what we noticed is that this trait is highly heritable. You might get differences from maybe from QTL, which are very dependent on the environment, but fruit feeding, which translates to the size of the fruit, is highly heritable in the sense that when you have a fruit in one site, you're going to have that fruit, maybe the same fruit with different sizes, but the fruit feeding will be there. So you can't have a shriveled banana and a good banana in another environment. So the heritability was very high for the trait itself, which is why we're confident of the QTL across the environment.
Okay, thank you very much. Thank you very much.
So Brigitte, my last question from my side is that what could be the support to advance even more the banana breeding program in terms of the genotyping?
We have a lot of things in the pipeline. As I indicated, we have so much in the way. So what we hope to get from support we need to get from you is the validation of this QTL in different backgrounds. That would be a big help so that you can decide on which one you are going to use in which background. So it's really good that you've been supporting us so far, what we've been doing, but we still need, and when we talk about this support in bananas, when I say, oh, this QTL has been verified, oh, validation can be done in 1 month. No, that's not works in banana. Phenotyping takes long. So when you talk about 1 year in bananas, like 2 months, like if 1 year, another cross, like 2 months in bananas. So we need longer term plan on support, not just 1 year.
Thank you. Bodo, you can unmute yourself and ask the question, please.
Okay. Good afternoon. So, yeah, thank you, Brigitte, for this further clarification on these works there. I apologize, this phone is ringing. Okay, it stops. Yeah, I have, how to say, well, would be several questions, but the most important one is probably it is not directly related to what you were talking about, but because you raised this issue, you talked about fruit filling as a trait. So now coming from other crops, maybe my understanding of fruit filling might vary from the understanding of fruit filling the bananas at IITA have. And it would be nice to get a clear definition of that. Also, when seeing all the bananas with their variegation of the domesticated forms as a continuity, because it is a blend of a few species, okay? Could we talk of one and the same range of one of the same trait, or should we talk of different traits? For example, the fruit filling trait in matoke might be a different one from that in plantain, because when you look at the fruit as an outsider, one could, without knowing about the differences between bananas and plantains, one could come to the idea that, okay, the plantains are just rectangular or triangular, angular fruit diameter, whereas the matokes are round diameter fruit. So maybe this is a difference, I don't know. And besides that, it was interesting to hear you talking about these numbers of SNP markers that you have investigated there. On the other hand, from a practical point of view, and also coming from other crops and QA, QC and marker application in other crops, what people tend to do is to reduce the number of SNPs needed to reach a certain information. When I hear 50 SNPs or something like that, to, you know, for QA, QC, one could easily think this is way too much, and therefore the study is still at the beginning. So maybe you can specify if you have some SNPs for some specific purposes or so. But for example, for this issue of contamination taking place during the pollination work, in sunflower, where I came from the company, we had 5 to 6 SNPs that we checked for that, and that was sufficient to reliably detect any contamination that was, you know, in a cross combination A times B. 20 or 30 individuals, seedlings, were tested, and when there was contamination, one would show up with a different marker pattern. And this was absolutely confirmed always in the practice. So it was not necessary to do this exercise for 55 markers or whatever. It is also a matter of cost. So, and in this connection, I was wondering, because you are talking about these things from your experience, I realize, you look at it in a more generalized way, but sometimes it would be useful to get the hard figures that are behind there. For example, you said the few hybrids that are coming there, or you said not so many hybrids coming in every season out of the banana plantain programs. So how many in fact are these? Are these a few hundred, or are they a few thousand? This should make a difference. So maybe for the future, a little bit more concrete information would be helpful. Thank you.
Thank you, Bodo, for your insight. I will try to remember everything talked about. When you talk about fruit feeding is, I mentioned it at the beginning of my presentation, how we define it. We define it as the presence or absence of pulp, which is what you see here. Whether it's rectangular or in triangle or either form, we look at the presence of the pulp and the diameter of it, because once you peel those bananas, the shape you see outside the shape of the peel, it's not the shape of the pulp. So when, I don't know if you have ever peeled a plantain, once you peel it, it's totally cylindrical inside. So what we're doing at the beginning of when we're mapping measuring the trait, we're measuring the pulp. So we're cutting and measuring inside, but this is a lot of work. So we're also doing other easy measurable traits to see if we can find a trait which we can measure in a faster way. That's how we came up with, if you just use a tape around the finger without cutting it, the correlation between the finger and the pulp is 0.99, which means actually the peel is not what is the biggest, or the peel does not cause a difference between, does not cause an interaction between the size of the pulp and the size of the peel from one genotype to another. So that's why you see these very high correlations. So I hope here you understand what you mean by fruit feeding. The next question you mentioned is about the markers 55. We don't do QC with 55. Those are for verification. Even for sunflowers or other crops, before you get to 2 markers, you start with many of them. You can start with 2 and hope that those 2 are going to work. When we verify 55, probably half of them are going to work for QC. For a QTL, in one QTL, we verify all of them, because at the beginning, we actually made the mistake of trying the most significant one. They all failed. They refused to amplify for some reasons. So that's the reason why we start with the optimum number of markers, and in the process of validation, we narrow them down to what we're going to use. So we don't use 55, and I can shed more light there. That's how it works. The other question, when you talk about numbers, I can give you numbers, but for the sake of time, to genotype with KASP markers at Intertech, the minimum is 4 plates, which means even if I send 2 samples, I'm going to pay for 376 samples. So that means if I have 1 plate, I'm going to pay for 4 plates anyway. So it's to my advantage that I have at least 4 plates. So depending on what the purpose of genotyping, I showed where we sent even more than 3,000 samples at a time, but it's impossible for one station to get, let's say, 16 hybrids in one go, from bananas, 618 bananas. We are going to be getting a lot from 2x by 2x for that, sure. But for the other, let's say for 3x by 2x, you talk about 15 plants. From 4x by 2x, they increase more. So that means when I talk about few samples, it's only few in terms of terms, but in terms of multiples or 4 plates for genotyping. Thank you.
Thank you, Brigitte. Thank you, Bodo. So we are on the time. If there is no more questions, I think we can say that we came to the end of the 8th seminar of this series. The agenda for 2024 is ready, so it will be shared exactly in the same website that we shared the agenda for 2023. I can say that we will have seminars very, very interesting in 2024, exactly as in 2023. So please attend. So I want really to thank you, all of you, for being with us along all this time, and hope to see you in February, that it will be the first seminar of the series of 2024. And wishing you all a great year for 2024. And for the ones that celebrate the Christmas, I wish you a wonderful Christmas. Thank you very much, and see you in the next seminar. Thank you.
Thank you, Anna. Thank you, everyone.
Thank you, Anna. Yeah, thank you.
Thanks, Anna. Thanks,
Thank
IT
you.
team. Bye.
Bye-bye.
Bye bye, Wade.
Thank you. Bye.